Okay, I'm gonna make this one for lab 11. I think the preview video on this one is pretty good though, so I would go back to that and kind of recheck if that's there. So essentially, gene expression, it's vital, it takes energy. So what we saw in bacteria was that the lactose digesting enzyme, that B-galactose or whatever, that's only gonna be on if lactose is around. So here's our DNA, and here's our B-galactose, we'll call that B-gal, and this is that gene right there. So RNA polymerase wants to turn this gene on and go. That means go right there. So we'll say RNA pol. However, it can't because a repressor, let's call it R, is always going to be bound to this gene to prevent this from happening. Because you don't want this to just be putting out enzyme all the time. However, in the case when you do get lactose coming in, lactose removes this repressor, and then you have RNA polymerase, and that's gonna cause B-gal to go up. And then B-gal can go and digest the lactose, get all the products from it. But remember what that does is that once there's no lactose, this repressor is gonna come right back, bind right where it was. No more RNA pol, no more enzymes. So see how that cycle kind of worked? Once the enzyme actually acts on its product, it's gonna get rid of lactose, the repressor is gonna come right back, no more gene anymore. So that's why it's a very controlled system. The two main chemicals that we used for our system were IPTG and I think ONTG. So they did two different things. They're mimicking lactose in both senses, but we just showed you lactose. So let's call this here, we'll just call this IPTG here. IPTG's role, that was just to bind the repressor, get rid of the repressor, and that equals B-gal is now on. ONTG was mimicking lactose, but at a different stage. Now, what this one was doing was saying that B-gal was now active after we added IPG, remember? Instead of lactose, we put in ONTG because when it was broken down, it released one kind of byproduct over here, but another that it released was a yellow color. All right, let's try this. I'm gonna mix green and blue. Is it gonna turn yellow? Nope, not very artsy. Okay, imagine that's yellow. So essentially ONTG was a signal that B-gal was out and processing lactose, or a lactose derivative like ONTG. So, two big ones, IPTG mimicked, it bound the receptor, turned the gene on. Once the gene was on, we added ONTG. ONTG was bound by B-galactose, digested, and we got our yellow signal. So upon receiving the yellow signal, we could confirm that the gene was on for that period. So remember we had all those increments of time. It was like 0, 5, 10, 15, and we pretty much we could see at the certain time point when the activation happened. And I think we actually observed that it kind of went like that. You know, the longer that IPTG was added for and allowed to be, the more B-gal there actually existed. Thus, the more B-gal was able to digest more ONTG. So that was the system that we used in that lab. And so these videos, yeah, if you've been watching, these videos are definitely a little more brief than usual. That's because I'm staying pretty broad right now, or I'm trying to. Email me, got things that maybe you're confused in the videos, because I might not know if I don't see it, and anything else extra you want.